Yeah, good afternoon, everyone. I'm Dr. John Kenodia, I head research department. I'm also an obstetrician gynecologist.
Okay, Dr. Kutoi?
Good afternoon, everyone. I'm Dr. Lydia Kutoi. I head healthcare quality division, and I'm also a gynecologist by background. Thank you.
And they do a very good job at quality. There's somebody who's very close to my heart, Mukwana.
Good afternoon, everyone. My name is Raheli Muhwana. I'm a midwife in charge of labor ward Kenyatta National Hospital. Thank you.
Okay, so Molly, sorry, we haven't met, but kindly introduce yourself.
Yeah, I wish we could be there today, but my name is Dr. Molly Blank. I am a biomedical engineering PhD and formerly the research director at Shift Labs putting the study together.
Okay, excellent. Nice hearing from you, Molly. Damaris Kimonge, please. Damaris seems not to have joined. So we'll have Shevin Jacob. I don't know, I hope I've said that correctly. And same for Shevin. And I don't know if Beth Kyoko has joined on. Oh, it's Kolko, sorry.
No problem. Hello. Yes, it is a pleasure to be online with you. Sorry that we can't be there in person. I am Professor Beth Kolko. I am the founder of Shift Labs and until last month was CEO. And I believe our new CEO is one of the attendees for the event. So we're very excited about the study and thank you for hosting this.
In my usual bad manners, I haven't introduced myself. So I'm Dr. Maureen Owiti. I'm the head of department for Kenyatta National Hospital, the obstetrics and gynecological department. And obviously this is something that is the core of our obstetrics. We have a lot of emergencies with the people with preeclampsia and eclampsia, and obviously oxytocin. We use a lot of oxytocin for augmentation of labor. So the findings are very interesting. So I hope everybody is going to be very well informed by the end of this. So I'd like to welcome, I don't know if Dr. Kinudia is the first presenter, Dr. Kinudia to start his presentation. And we'll also request, there's one of our medical officers who will be co-chairing with me,
Vivian Manyeki.
Dr. Vivian Manyeki, if she can also assist to co-chair, kind of also like having a few issues, but we'll be with you through the presentations. Thank you, Dr. Kinudia, over to you.
Thank you, Dr. Owiti. Dr. Kutoi, you are ready? And you need somebody to move the slides?
Yes, I'm ready, and we are ready to move the slides too. We can begin. Great. So we are glad that we are able to share the results on the evaluation of DripAssist, which is an innovation for infusion. And in our case, it was for magnesium sulfate and syntocin, oxytocin. So medications during labor, we realized that medication access is essential in addressing preventable maternal death. And some of the medications that I've mentioned are considered to be signal drugs for ensuring every maternity unit has them, because they literally prevent death when they are used appropriately and optimally. So drug delivery method, when we're considering on how to deliver the drugs, we consider the patient needs and their condition, and then also on matter safety, which is the best route, and is it being done in a manner that we ensure that there is no risk of infection, and then also in what setting, is it at home, is it in the clinic, and who is doing it? Are we having what we call a trained skilled worker who can offer emergency obstetric care? And then also, how are we able to confirm accuracy depending on the kind of molecule that is being used and the form that it is? and also on matters, methods of delivery, we look at the gadgets that we have, that is the equipment, are they available and what is the cost, especially when you're talking about the infusion approaches that we have, whether be it by the normal drip stands that we have that use gravity, or is it a pump? So how available are they? And then also what is the speed of mechanisms of these drugs, and how dependent is it on the speed that you're giving, and is it within therapeutic and safety levels? And in all these approaches also, what man hours are we giving this staff to concentrate on that gadget vis-a-vis monitoring the patient, how the baby is, the blood pressures, and at the same time being able to regulate and give optimal doses of these drugs. And in our case, it is about oxytocin and magnesium sulfate. So in terms of accuracy, when we talk of dose accuracy for PPH, we have misoprostol and oxytocin. Oxytocin 10 international units IM is the standard for management of third stage of labor. And so it is a safe drug and it saves life. And if you're not able to give that 10 international units IM, or if there's bleeding, then the option of 20 international units in an infusion drip is also an option that we take. So it's been known that each of these options have their own challenges, especially misoprostol. At times it's difficult to do the dosing. However, the advantage is it can be done given orally, or it can also be given parectally. However, on matters dosage and accuracy, misoprostol is more challenging than when you come to oxytocin. But now oxytocin being delivered by drip, then there are also challenges to do with how much, like if you're using it for induction of labor, just being able to calibrate and to ensure that you're titrating it correctly also gives us the challenge of just the time and having the correct optimal doses being delivered. And that has been confirmed in many studies. And these studies are the ones that give us the level of evidence for WHO to give this intervention for using PPH, for using third stage of labor routinely. And so we continue looking at ways to optimize the challenges that come with any of these drugs, so that there's optimal delivery of drug when you need. Going to magnesium sulfate, which is our other drug of interest, is that magnesium sulfate also has been said to have some therapeutic safety windows, and in case it is given rapidly, then you can get into the toxicity, which can lead to respiratory arrest and cardiac arrest, or even death, because we know that it leads to depression of respiration. However, magnesium itself is a life-saving drug, evidence that it is a signal drug to be there in every other maternity unit to be used for hypertension, pregnancy-induced hypertension. And so accurate optimal delivery of magnesium sulfate is important. Just like the previous drug we've mentioned, when given through the drip, there is the challenge of ensuring that we're giving within the therapeutic window that is allowed. Indeed, IV medications are essential, as we've mentioned, in providing treatment and in preventing maternal death. And accurate administration of IV medication will save the patient. And we also have to look at the harm in case we are out of the therapeutic window. What will happen to the mother who is in labor in terms of the contractions and the fetal status, and what will happen to this other a lady, either who doesn't get optimal magnesium sulfate or gets to the levels of toxicity. So it is essential that we provide this treatment, but within the therapeutic levels that are acceptable. Okay, these are some of the devices we have for infusion. The normal is the one that is used. We use gravity, the first one on my left. We use the normal infusion bag, and we clip it at the number of drops. We titrate it to the number of drops that we need per minute. That has its challenges because at times we don't even have the correct tubings. At times they slip. At times, now with the usual labor movements, because the mother may bend their hand, the level of the bed, and many other factors could affect that. Then we have the syringe pumps, and that are various approaches. There's the one that is in green, and then we have this other one, the larger volume pumps also. You'll see them in theater, in CCUs. And all these also have cost implications. Our study did not really look at the cost implications, but the truth is some of them are fairly expensive. So the challenges of accuracy, that those that are gravity related, or even the pump itself could have its own challenges. But using the gravity mechanisms, what we commonly see in our unit, you can see that the either too fast or too slow, the error rate is about 39%. That is quite a high rate. It's almost going to 40%, or more than a third of all those who on those drips, there's an error in the rate in which the drug is being delivered. Then even those with a pump, also it is not usually 100%. In hospital settings, it's about 23%, even with a pump, either on matters of how it was calibrated or just some failure within the system. This is a picture just to show us the various dynamics that could affect a patient in bed receiving fluid using the gravity approaches, that is the normal infusion pump that we have. You can see the atmospheric pressure, even the level of the infusion, the drip stand, viscosity, whether the patient is moving, the patient's blood pressure, and even the kind of tubings and the kind of clip that we have. The roller clamp slippage at times could happen depending on the quality. That's why in many facilities, there's a TEC to look at these that are being delivered. And in case there's a challenge with one, it's reported so that we don't take such. So the roller clamp slippage is also a common challenge, and even the position of the bed and how the patient is angled will affect the speed of flow. So coming back to our study, that was the background. In our study, our objectives were to understand the impact of a drop counting device as a substitute for manual drop counting in a maternal health setting. And our specific objective, number one, was to see what our intervention was. It's called a DripAssist device. It was to assess the effect of the DripAssist device on the accuracy of the prescribed dose and infusion rate of oxytocin and magnesium sulfate. And so for that, we were comparing before and after, and the method section will be able to elaborate on that. And our second objective was to look at the frequency of the side effect associated with the intravenous gravity infusion of oxytocin and magnesium sulfate. So we're also comparing the side effects that are common in patients who are receiving oxytocin and those who are on magnesium sulfate, comparing those without the DripAssist and those during the intervention with DripAssist. So thank you very much. We will go to the gadget, and I will invite Beth to continue and share about the gadget that we used for this intervention. Beth, you're welcome.
Thank you very much, Lydia. So can we go to the next slide? Thank you. So what I'd like to do is share just a little information about the DripAssist Infusion Rate Monitor device. So this is a device that Shift Labs developed. Shift Labs is a company based in Seattle, Washington, in the US. And we developed this device really based on work that I had been doing at the time as a professor. I'm also a professor at the University of Washington, and spent time with clinicians in different parts of the world. And one of the common themes that came up in that work was the challenge of gravity infusion. And really for all of the reasons that Dr. Otoyu was just describing, with that there are so many dynamic aspects to a gravity infusion. So when we started developing the DripAssist Infusion Rate Monitor, our goal was to create a device that would be versatile enough to work in really any kind of setting and would be robust and very easy to train on as well. Wouldn't need a lot of instruction. So the device itself is about the size of a cell phone or a deck of cards. It runs off of one AA battery, and it was designed to work with any drip set. So there's 4 sizes of drip sets around the world. Some countries only use 1 or 2 sizes, but we wanted this device to work with any size that a facility would have. So when you first turn on the device, you have the opportunity to tell it what kind of drip set you're using, what size, whether it's a 10, 15, 20, or 60. And the way the device works is it has some infrared detectors that watch for drops, essentially. So as soon as you start an infusion, the device will immediately show you the flow rate information, and it will also show you the total volume of fluid that has been administered. And it will show the flow rate information in either milliliters per hour, as you see on the screen here. It can also show drops per minute. So depending on the kind of infusion that you're conducting. And let's see, so it runs off a 1 AA battery, works with any drip set. So the infrared detection senses the drops, but the device has software on it. So there are algorithms on the device that take the flow rate information, or take the drops rather, and sort of process that, eliminates any sort of light interference or potentially double drops. And so it kind of creates quality data out of the drop detection. So in laboratory testing, the device has been proven to be 99% accurate as a drop rate detector. So in counting drops, it's 99% accurate. As I mentioned, you can see drops per minute, milliliters per hour, or total volume administered in milliliters. There is also an optional alarm that will alert when there is a significant flow rate change. So unlike an infusion pump where you would initially dial in a rate, the DripAssist works a little bit differently. What you do is you start your infusion, and then once the screen tells you that you're close to the rate that you want, that's when you would hit the 3rd button on the device, which is an alarm button or an alert. And then it's a set point, okay, for your infusion rate. And if your patient moves or the bag gets lower, something happens and it changes the rate, whether it goes too low or too high, that alert will sound. So as I mentioned, we built the device so it wouldn't require a lot of training. There are no sub-menus. You're looking at this device with me. You can see there are 4 buttons and that's it. That first button on the left turns the device on and off. That second button is where you would select the drip set that you're using. The 3rd button, as I mentioned, is that optional alert. And then that 4th button tells you whether it's milliliters per hour, or you can see milliliters per hour drops per minute of volume. Just toggle through. If you'd like to see the device in action, you can go to YouTube. I should have put the URL on here. I can put it into the chat, but it's just youtube.com and then /shiftlabs, it's the name of the company. And we have a series of videos, including a 1-minute video that walks you through all of the features and functions of the device. So as I mentioned, it's quite small. It's about, I think it's 108 grams, I believe. Lightweight, and it will fit on any drip set. Can we go to the next slide? And on this slide, what you see are, well, those 4 buttons I just described. But I also wanted to just address, we're in the middle part of the slide where it says insert drip chamber. And there's sort of 3 blue pieces there. The piece on the right is a rotating cam, so it moves up and down. And so when I said that it will fit any drip set, that rotating cam is what allows the device to adapt. The device never needs any calibration. So unlike an infusion pump, once it's deployed into a ward, it never needs maintenance or calibration. It's easy to clean. It's resistant to water, to fluid. And so it really is just designed to be able to be sort of taken out of the box, used, and the only maintenance required would be changing the battery. The device is cleared by the US FDA. It also has a CE mark. It's manufactured in the US. And this is our first clinical study we've done. We do have customers around the world who have used it, and we have clinical case studies, but this is our first actual clinical study data. So, and that's really, if during the Q&A, I'm happy to answer any questions. And in the chat, I'll go ahead and put that URL if you'd like to see the device in action. There's some short videos there. So thank you very much. And I think now I'm going to hand it off to Mukwana. To talk about methodology. Great.
Yeah, this was a quasi-experimental pre-post study, which we compared the outcomes between the current practice in our labor ward, that is before the DripAssist was introduced, in the next phase, the second phase, which was the post phase. Yeah? And during this time, we managed, in the pre-study, we had 500 participants who were enrolled, and the post-study had 316 participants. And then the study site, as we said, was at Kenyatta National Hospital, a labor ward, which is a very busy maternity ward, and receives patients from other referral centers and also walk-in patients. And you find the patients we needed for this study are quite a number in the unit. You find they're about a third of the patients who are admitted in the unit. So a study population comprised of pregnant women who had been prescribed for magnesium sulfate for pre-eclampsia and oxytocin for augmentation of labor. So what happened? We had study nurses who had been trained. We had recruited study nurses who were very independent from the rest of the nurses in the labor unit. So they had been trained on the DripAssist, so they would get consent. So we'd get women who are able to give consent. Remember, we were getting patients who have eclampsia. So those ones who are conscious and were able to give consent will get their consent. But those ones who were not able to give consent, we would not recruit them into the study. And the ages were between 15 and 50 years. So we had these study nurses, as I told you, had been recruited and were independent from the labor ward nurses. So they went through a training, almost like a 2-week training to familiarize themselves with the red cup and with the gadget, the DripAssist gadget. So they would consent these women on admission, and they would only start collecting data when either magnesium sulfate had been prescribed or oxytocin. So it had to be a prescribed drug in on the treatment sheet. So they would collect data, they would monitor these women hourly. And during the monitoring, they would take the vital signs, which is the blood pressure, the temperature, the pulse, respiration. And now this is for all the patients on oxytocin, and and magnesium sulfate, and also the fetal heart rate. Then for the patients on magnesium sulfate, they would take the patella reflexes and also the output, the urine output of these patients. So at any given time, we'd have 3 to 5 women who were being monitored. And then, and this is the constant burden, the number of patients we wanted to be monitored. We didn't want them to have so many patients, then they're unable to monitor these patients. So when the data capture was complete, maybe for the 5th patient, then the next patient would be fitted in into the study, the one who met the inclusion criteria. So the other data that was collected hourly was the weight of the bag from the time the infusion was started to when the infusion was stopped. And when it would be stopped is for the magnesium sulfate is after 24 hours. And then for the oxytocin is when the patient had delivered. And sometimes it would be stopped, maybe interrupted when there's a radiological exam that needs to be done on this patient. I would invite Molly for the next part of the presentation.
Thank you very much, and good afternoon, everyone. It's a privilege to be the one who actually gets to present these results after all the incredible hard work by the Shift Labs and Kenyatta National Hospital teams. So in these slides, I will share the results of the study that address both the impact of the DripAssist on the accuracy of the bags, the infused bags, and on the frequency of side effects. I would also like to acknowledge before I really get started that the data processing effort was shared between myself and Damaris Kimonge, who did the majority of the statistical analysis and tremendous work preparing these results that I get to share with you today. Another important thing to note is that all of the results reported today focus on our findings around magnesium sulfate. Because of COVID-19, fewer patients were indicated for the oxytocin for induction. And for our pre-post analysis, we only reached enough patients to have a significant conclusion for the patients presenting with the preeclampsia or eclampsia. So we are still working with the data in anticipation of publication, but these initial findings today are focused on magnesium sulfate. So this first table summarizes the demographic information of the women who were consented into the study. There were a total of 274 magnesium sulfate patients in the pre-intervention group and 260 in the intervention. And people can ask more specific questions about this, but the largest change that we observe in these populations is the proportion of patients who had college and university training, although I don't really have insight into this fact. Largely most of the other factors spanning occupation, marital, and education level otherwise are pretty much unchanged. Can I get the next slide, please? So when it comes to the accuracy findings of the study, I think if you walk away with remembering only one thing, this is a very good slide to focus on. This figure presents the accuracy of the average rate for an entire infused bag of magnesium sulfate. So here we define the term accurate to be as within a 10% margin of the prescribed rate. And the use of the DripAssist in the intervention period, and this is the component of this figure that is circled, the DripAssist almost doubled the number of bags that were infused accurately. So with the DripAssist, the number of bags that were within that 10% of the prescribed rate went from a little over a quarter of the bags to a little more than half. And we find that this is a statistically significant impact of the DripAssist on the overall accuracy of the infused bags. And here in the figure, you'll also see we broke out the number that were too slow and that we're too fast. So less than that 10% and greater than that 10% difference. And the most significant change happens in reducing the number of infusions that were administered too quickly. So more than, this may be something to keep in mind as we get into the effects of the intervention on the side effects that we observed. Can I get the next slide, please? So now that we've summarized sort of the clearest finding of the study, we return to a snapshot of the raw data. So here on the vertical y-axis, we see the difference between each hourly measurement of the rate in mils per hour and the prescribed rate for that hour. So for each hourly measurement, what is that error? And then on the x-axis, the horizontal axis, we have the date that measurement was taken, and shown as each hourly measurement taken for the duration of the study for magnesium sulfate. And I wanted to point out two things here. The first is that the last patients in the pre-intervention group were consented in February of 2020. And that corresponds with the beginning of the global spread of COVID-19. We've done our best to consider this for the analysis, but besides the impacts on the consenting of the oxytocin population, we have found no other effects that significantly impact our conclusions from the study so far. The second point that I want to point out that we see here in the raw data that we go on to verify in our analysis is that the intervention period, the blue points on the right, appear to reduce the variance of the error. So fewer hourly measurements in the intervention period result in really large differences from the prescribed rate. So that data is just a little bit tighter. And the key results that I present in the remainder of these figures and slides are actually based on the average rate for the entire infused bag, but it is important to note that we observe all the same conclusions on an hourly level as well. So next slide, please. So this slide is mostly to demonstrate that the considerations that we took while approaching the data. The main point here is when I say there's a statistically significant difference between the pre-intervention and intervention groups, we use tests that assume that the data had a normal distribution or the sort of classic bell curve. And for both the pre-intervention and intervention periods, these straight lines indicate that our assumption of a normal distribution could be confirmed. I don't want to spend too much time here because this is mostly sort of checking a key assumption about our data, but I'd be happy to return to this if there are questions. Next slide, please. So here we show two histograms comparing the frequency of the percent error for the intervention period, which is that top sort of purple blue, and the pre-intervention group below in that red. And the number of infused bags, the frequency of that percent error is shown on the y-axis, that vertical axis. And on the x-axis, we see a range from negative 100% to 100% error relative to the prescribed rate. And there are some bags that were infused even more than 100% faster than prescribed. There's not a bunch, but there's some data that's not shown on this figure. And here again, we see what we observed in the raw data, that there's a much tighter distribution in the intervention period with the use of the DripAssist, with many more bags that are close to that sort of 0% error exact infusion mark in the middle. And because there's such a large spread in the pre-intervention period on the bottom, and the mean value is sort of erring fast, I think it's more meaningful to compare the median values, which were both a little slow, which we see based on sort of the peak of that curve. And so there's a little bit of a change in the median by about 3%, but the variance between the two is really significantly impacted. So we did find that there is a significant reduction in the variance, that sort of spread, that range in the pre and post intervention periods. Next slide, please. So finally, we have here the findings for these side effects that were observed in both the pre and post intervention phase. And here we go actually back from the bag averages to each individual observation as these side effects were taken at each individual hour, each time there was a measurement. And what percentage of all of those observations included any of the side effects listed. I also want to point out that not listed here, we did observe loss of consciousness or the occurrence of seizures, but there were not a significant number of each of these in either the pre or post groups, so we were unable to make a comparison between those. But we found that for headache, stomach pain, and nausea associated with magnesium sulfate, they had significant decreases, and the largest changes occurred for nausea and stomach pain. So this is a pretty significant finding, and it's among the first that I'm aware of that really definitively connect dose rate with the incidence of side effects, especially based on a device intervention that was the DripAssist. All right, next slide, please. So to conclude here, the data indicate that we can confirm the two hypotheses of this study that we went in to evaluate our goals here, although it was only for magnesium sulfate, again, due to the consenting issues associated with COVID-19. But first, we find that more infusions can be administered accurately with the use of the DripAssist device. There is a significant impact, and that for certain side effects, we did observe a decreased frequency of those side effects when the DripAssist is used. So with that, I want to thank you, and I'll hand it back to Raheli Makwana for the final words.
Thank you, Molly. And also, I want to thank everyone for participating and coming to listen to this webinar. And with this study, we had several people who put in a lot of effort to ensure that the study was complete and it ran smoothly. And these are our study participants. Our study participants were cooperative. They gave us consent. They allowed us to collect data from them. I also want to thank the study staff, the nurses, and especially who worked, tirelessly in the labor ward, and they were able to mingle with the other staff in labor ward, and they were a great deal of help in the labor ward, and they were very efficient. I also want to thank the study investigators who made sure that the study was running well, and anytime we needed their help, they were there for us. I will not forget our institution, Kenyatta National Hospital Management, through the Research and Programs Department, that gave us, they're the ones who facilitated this and also allowed us to do this study in the hospital. Despite the busy labor ward, they allowed us and other studies going on in the labor ward. I will not also forget Shift Lab for the innovation that I believe most of the people really, according to when I see the question and answers and the chats, are eager to use the device in their facilities. This study would not have been possible without the funding agencies, that is Saving Lives and Births, USAID, KOICA, Bill and Melinda Gates, Grand Challenges Canada, and the others who made funding available for this study. Thank you very much. And back to Dr. Kinuthia or Dr. Maina. Moreno, sorry. Yes.
Yeah,
Thank you.
Thank you, Laheli, for that. We'll hand over now to Dr. Vivian Manyeki and Agnes Karume to moderate the rest of the session.
Thank you all. This is Dr. Karume speaking. We will thank our panelists for that elaborate presentation and for the work that has gone into this study. For our attendees, thank you for joining us. And what we are discussing today is just evaluation of the drip scan to increase safe access to magnesium sulfate and oxytocin. And you've seen the impact it has had on both sides, on both the healthcare workers and the patients themselves in terms of the outcomes and reduction of errors when it comes to monitoring these medications. And we'll now go into some of the questions that have been asked in regards to this study. And one of the things that is being asked is about the availability of the device. We've seen how helpful it is, how about the availability of the device and also the cost implication that may come with it. And this, as any of our panelists who may want to respond to this, maybe Dr. Owiti, you can make a comment on this.
Hi, this is Beth Kulko from Shift Labs. I can respond to that question. So we have spoken with various potential distributors in Kenya. The device is registered with PBB. And I would say that the availability is sort of up to your colleagues in Kenya, but our hope is that this data will help to drive awareness of the device. And certainly if clinical facilities are interested in using the device and purchasing the device, it will absolutely be available. And then in terms of the cost, that's hard for me to say in terms of, I know there's import taxes and things like that once it gets to Kenya. But you know, $100 to $200 is sort of we would hope that that would be the maximum. So I'm sorry, I don't have a more precise answer, but I certainly hope that on the heels of this study, we will be able to have more collaborations in Kenya. Thank you.
Thank you so much for that, Coco. And indeed, it will be lovely to have it widely available. I just see it even as we continue in the questions. I'd like to invite even some of other panelists to with us. Dr. Irungu Kamau, if you can make a comment on just even the general presentation before we delve into the rest of the questions.
Thank you so much, Agnes. My name is Dr. Irungu Kamau. I work at the Division of Patient and Health Worker Safety at the Ministry of Health. And the division lies within the Directorate of Health Standards, Quality Assurance and Regulation. And maybe mine is just a few comments that, of course, as a ministry, we appreciate technology. And since we are aware, of course, technology at the end of the day improves healthcare, efficiency, quality, as well as safety of both the patient and the healthcare worker. But of course, on the other hand, we cannot fail to say that the same technology sometimes may introduce errors and adverse events. So mine is to commend the study and it has shown the value of the DripAssist technology in the delivery of both the oxytocin and magnesium sulfate. But of course, at the end of the day, we need to be able to balance the good and any adverse events that may be related to the device. So represent the division, I'm saying we are happy, and I'm happy personally because we are really advocating for any measure that may improve the safety of both the patient and the health worker. So those are just my few comments and remarks, and thank you so much. Thank you.
Thank you so much, Dr. Kama, for joining us. Dr. Shevin Jacob here also with us, if you can give also some of the comments on the presentation and on the work being done.
Yeah, sure. Hi, everyone. Yeah, I didn't get to introduce myself earlier, but yeah, my name is Shevin Jacob, and I'm an infectious diseases physician at the Liverpool School of Tropical Medicine, who's based in Uganda. So I've been doing research here on severe illnesses, particularly severe infections like sepsis and viral hemorrhagic fevers for about 15 years. And it's really, and I might have been an advisor to Shift Labs and one of the co-investigators on this study. And it's really exciting to see the close of the study and this preliminary data as presented. I thought that it might be useful. I can see in the over 1,000 participants that there are a number of clinicians who are listed here, and I thought that it might be useful to sort of think hear about other use cases for the DripAssist. I am also a consultant for the World Health Organization and have been part of the response to Ebola outbreaks, most recently in the Democratic Republic of Congo for the 2018 outbreak there. And as some of you may know, there was sort of during this outbreak, it was the first introduction of therapeutics that were initially being done under sort of a compassionate use protocol called MURI, and then eventually evaluated in a randomized controlled trial called the PALM trial. You know, in managing patients in an Ebola treatment unit, as many of you have seen or experienced, it requires that you're wearing pretty restrictive personal protective equipment. And the time that you can wear that equipment is limited. And yet you have critically ill patients who require life-saving therapeutics, whether that just be intravenous fluids or some of these new agents, the monoclonal antibodies. And it was in the DRC during the recent outbreak where drugs like ZMapp, which were one of the therapeutics that were being evaluated in the PALM trial, that required sort of administration over a 4 to 5 hour period, were being administered with the use of the DripAssist. And Professor Kolko sort of was able to walk you through some of the features of the DripAssist. And one of the key features, I think, and particularly in this use case, is the alarm feature, where you sort of have your ability to set what rate you want to administer your drug, but also when there is sort of variation, either too high or too low with respect to that rate, the alarm goes off. And this is critical when you're managing, you know, and it may not just be an Ebola treatment unit, but you can imagine on any busy ward where you have drugs where it's important to administer for life-saving reasons. But at the same time, you may have a number of patients that you're trying to care for at the same time. So having a device that's able to provide you with an alarm when there is an aberration in the rate, or when you've completed sort of the treatment, is quite useful in terms of efficiency, but also in terms of being able to manage patients in the safest way possible. So that was my experience in DRC using sort of administering these different therapeutics and having sort of the DripAssist to give us a little bit of reassurance that what we were giving was being administered accurately. And when there was sort of variation because of the viscosity of the treatment, et cetera, we were aware of that and made aware of that because of the features of the device. But I'll stop there. Over.
Thank you so much, Dr. Sevin, for those comments all the way from Uganda. We really appreciate. And even as we go on, I'd like to invite also Dr. Joyce from Mishre, who's part of our panelists. You can also do a comment on the study.
Okay, so good afternoon and thank you for having me here. I work at the Ministry of Health in the Division of Research and Innovation. And I really enjoyed listening to this study. So from my angle, first, when we are speaking from this side, not of the ministry, we're always asking ourselves, okay, can this be scaled up? What is the scalable level? What does it mean? Cost-wise, how, I mean, those are the questions we always have at the back of our minds. So first and foremost, we are happy that this innovation meets several SDGs. We get really excited when we hear that our local institutions are collaborating with other institutions out there. So kudos to Ken H and to Dr. Kinuthia and team for working with, for being in great partnerships like with Shift Labs. Secondly, any issue to do with mothers is very central, I mean, to the Ministry of Health, mothers and children. We do know a lot of issues really affect those two subpopulations. So that's the other plus for this. And then we are really into innovations that are scalable. So when we think about what has been presented, there is the whole issue of awareness. How do we get other people to know what this study is talking about? So perhaps the team can consider doing an evidence brief so that we now are able to see exactly what was happening in the study. It's a précis kind of brief that really tells us everything that they learned in the study. But for it now to move to a level of policy, questions that that I can see, some of them I can see in the chat, others have been mentioned by the panelists, especially my colleague from MOH, are the issues to do with costs. So what are the cost implications if we are to scale this up? Definitely, this is a device that can be used right up to the dispensary level, or rather the healthcare the health center level. But what does it mean cost-wise? If you now calculate that it's about 100 to 200 US dollars per device, and at any one time, you know, if you're looking at in the maternity wards and in the children's ward, how many drips do you see hanging? So even if you take a modest number of 10, what does that mean at that level? And then once you buy, the issues of batteries, are they rechargeable? Is it something, because we find even when we are rolling out things like e-health, we find simple things like asking a facility to cater for costs like antivirus. You will find a computer seated somewhere in the corner of a facility, and when you ask why it's not being used, you're told, no, there's no antivirus. So to move from evidence to policy brief, perhaps I would encourage the team to maybe now do this study, but in a real life setting, not the way like you've done clinically. Controlled, not the way you've done in a controlled setting, but now dare to go to and see how this device would work at lower level health facilities, and really even do the costing for the HR to be trained, and for what does it mean when you now have the DripAssist devices at that level, so that now when you're doing a policy brief, those are the questions that the policy makers are really interested in. And then lastly, issues to do with now, if you do have an evidence brief in terms of awareness also, we do have a resource at the Ministry of Health called the Kenya Health and Research Observatory. You can send us the evidence brief. I'm sure Dr. Kinuthia can give you our emails, and then we can upload that onto the observatory. So that gets read by many, many more people, including the CECs and the county directors of health at our county, at our various counties, and raises awareness. But I can tell you, before now people make the decision of, can we have this in our facilities? There are further questions that need to be answered, and I think I've belabored on that. Otherwise, great study, and we like innovations, and innovations that specifically can work at all levels of facilities. So thank you.
Thank you very much, Dr. Joyce. Indeed, those are very valid concerns. We need to look at the scalability, the cost issues, and how do we even roll this out for the benefits of our patients. And we'll get into that discussion, to a bit of that discussion too. Dr. Ankunda Karisa, I asked if you can also make a comment. Karisa, Ankunda. To get back. Now, as you see, some of our participants, thank you for joining us. And some of the direct questions that go into the patient care in regards to the management of labor will not be able to get into those ones today. Today we are discussing about the drip assist that is assisting us in the monitoring of our patients as we give some of this medication. And in this particular point, you're talking about magnesium sulfate and oxytocin for the safety of our mothers. One of the questions that we direct to Mukwana is, will you give us an evaluation as the nurses, the study participants as the nurses were monitoring this medication? How was their experience? What are some of the experiences? Was it easy to use? What are some of the comments? What are some of the challenges you experienced during the rollout of this study? Just in brief.
Yeah, it was according to the midwives working in labor ward. They interacted with the device because, as I told you, the other study nurses were in labor ward, and they felt like it was light. As Beth said, it's the size of a mobile phone, so it was not as bulky as the other gadgets that we use to regulate fluid. And then you see, this is with the number of patients, the patient load. You realize that with the drip assist, it had an alarm, so it would alarm you in case the flow is not what you had set. So they also had a chance to interact with the device, and just to familiarize, though they were not the ones collecting the data, but we had to incorporate them, and they had to know how the device works. But most of them, in fact, they have been asking, when do we get this device? Because if you realize, with the other fluid pumps are very expensive, and you cannot afford to have a fluid pump for more than 30 patients being monitored, either on oxytocin or magnesium sulfate. So it's something they're looking forward to use, and I'm hoping the hospital will procure a few for use in the unit. Thank you.
Thank you very much for that. In our chat boxes, you can be able to see some of the website, and also a link that has been posted on where we can get even more information on how to use the drip assist. There's even a YouTube video from Shift Lab. Maybe KOICA, you can just tell us a bit, though you mentioned in your presentation, on just general views of the drip assist and how to monitor the drip using
I'm so sorry. I wanted to make sure that I heard the question. Just how it's being used to monitor drips elsewhere?
Yes, just I mentioned, which was part of your presentation, for how it's being used to monitor and how to use it generally.
Oh, yes.
You have the
Sure, of course. So in the US, the DripAssist is mostly used for outpatient care and in smaller clinics. It's used to administer a broad variety of medications. Very often it is used for monoclonal antibodies. As Shevin had mentioned, the device has been used in Ebola treatment units for monoclonal antibodies, but we also have in our health system a lot of home infusion done for chronic conditions that use monoclonal antibodies, and DripAssist has become a popular choice for those settings, in part because of the comments from I think it was Dr. From the ministry, in terms of staff training. So it takes about 5 minutes for someone to learn to use the DripAssist, and it's a device that it doesn't have the same kind of problems in the field that infusion pumps often have. So it's really useful in settings where you maybe don't have your biomed department close by. And it's also been used by a variety of international organizations, by WHO. I believe that WHO has largely procured for Ebola treatment centers in the DRC outbreak. Let's see, and then in the US, in smaller hospitals, it's been used for chemotherapy delivery, smaller clinics that don't have access to the expensive drip sets that would be used for pumps for chemotherapy delivery. So they've used DripAssist for that. It's used for antibiotics, a broad variety of medications. And the feedback that we get from nurses is one of the reasons they enjoy using the product is because it makes them feel sort of in control of the gravity infusion. And I saw in one of the questions, someone asked about the how the device can be 99% accurate, and but then the findings about the accuracy of the bags was that 52 or 53% numbers. Those are two different measures. So the 99% accuracy is the device's ability to measure drops. But once we start being in a clinic and there's multiple patients and demands on time, and then gravity infusion itself is quite variable a process. And so I think that Molly is answering that question in the chat, so I'm going to let her do that. But just to return, the nurses indicate that being able to feel sort of confident and in control of a variable gravity infusion is really important to them. So those are some of the settings where DripAssist is used, and some of the medications. And I know that I'm going to just take this opportunity to just say thank you to the team at KNH. And I also want to acknowledge that the study was done during the COVID outbreak, and it was not easy. And just a huge thank you for the Herculean efforts on behalf of the team. So happy to answer more questions as they come up. Thank you.
Thank you so much for that, Professor. Molly, maybe you can also add on the question regards to the specifics about someone says, yeah, this is an interesting and useful devices study. How else we determine the significance level, and just I mentioned also just a bit on the accuracy too.
Sure. Yes, I was just typing out this answer. Thank you. I think so. This is a summary of our preprint results. We do anticipate moving this into peer-reviewed publications. So this is a little bit preliminary, but for any claim that I made about a significant result, we do have p-values associated with these. We used a threshold of 0.001 for our p-values. So we didn't put those on the slides today, but for any claim, please be assured that there is a test behind that. And we do have those thresholds. And as we move into a publication, we'll be sure to include all of that information. But I appreciate the opportunity to provide that assurance.
Thank you for that. As we even come towards the end, as you're talking about the safe access and evaluation of the drip stand, we'd also like to look in at Dr. We'd also like to Dr. Kutoi, you can tell us a bit more, looking for maybe you can tell us a bit more. Sorry about that. You can tell us a bit more about how we can even scale it up from. We've already talked at the maternity wing, and we did the study, and how from our setup, we can even have it in more institutions or roll up this intervention.
Yeah, sure. I think just going back to what we got from Ministry of Health is we look at matter scalability, and scalability will be about the cost benefit that this device can bring, and then also just the aspect that it releases more time from the midwife, because at times if you have a very busy maternity and every other person is on oxytocin, it makes care quite heavy. So I think, as we've been advised, it is now about going to the natural conditions, or rather going to the natural settings in the other facilities. As you realize, it's been used in Ebola centers. And so those were places that had special effort. So in the US, it's been used widely, but I think in our setting, we propose to go to that next step. So our study did not involve those plans, but it was good enough to prove the concept that in Kenya, this can also be used to reduce the inaccuracies, and it is safe, that there are no untoward effect or increased side effects of the medication because of the device. So I think that's why we've been advised to do a policy brief, which now comes with recommendations. And as any product, even as you go out, the pharmacovigilance group, I think from the Pharmacy and Poisons Board, they regulate both the medications and the equipment. So also it still goes under watch if and when we go there. So I think we hope doing the policy brief will give enough clout that we can move that direction. There was a question on how it is used in terms of cleanliness. Mukwana will comment about that, so that we know that also when you get, it's one thing to have it and another thing to maintain it. So I think as a parting shot, she will comment about how much she depended on biomedical to come and do anything, and how she took care of the device, and how many she needed for that period, because this is not disposable. Yeah, thank you.
Okay, for how to clean the device, we didn't need to like immerse it in any disinfection. We just needed to wipe it with a disinfectant, since it was very far from the patient and hanging on the infusion. Yes, and then since it's using a battery, you don't need to use a lot of disinfectant because you will interfere with the device itself. Yes. Yes, and then as you know, every gadget needs to be calibrated. But for us, we didn't need to involve our biomedical. But for one or two gadgets that had a problem, had to be looked at. But we didn't have any problem with any of the gadgets used. We had enough gadgets to monitor at least 5 patients at any given time. Thank you.
Thank you very much for those comments. And even as we wind up, I think that most of our questions have been answered in regards to the accuracy, how we can be able to use this in other setups, the reusability, and even some aspects of how to use it. There's a link that has been sent to our chat box in regards, if you'd like to know more on how to use the device, you can check that on YouTube and you'll be able to know how to use it. For now, we are going to go into our last round of just comments from our presenters. Just comments from our presenter in regards to the use of the drip stand, even as we come towards the end of today's presentation. So maybe we'll start with you, KOICA.
Thank you. So one last comment about the costing before just a summary comment. I love this question, and this is something that we've worked on at our company as much as we can academically. And, you know, we've really emphasized simplicity of training as a way to reduce scale-up costs, being able to use rechargeable batteries, things like that. And certainly as manufacturing scales up, the price of the device drops way down. So I would love to continue that conversation about what scale up looks like. Since we are winding down here, and I'm going to sort of restate my thanks. It has been just an honor to work on this project with the team at Kenyatta National Hospital, and Dr. Knuthi and I have known each other for a few years through the University of Washington, and this is the first opportunity that we've had to work together on a research project. And it's been really, since I am a researcher as well, watching the commitment of the team through the challenges of the past year has been inspirational. And I'm very excited to say that we now have this clinical evidence about a product that we made years ago and have had great faith in. And I want to thank the whole team for their collaboration and making this study a reality. Thank you.
Thank you very much. We'll now have Dr. Molly Blank. You can also give us your comments.
Yeah, thank you. This has been a great privilege to work on this study. I would like to thank the clinical leadership team for helping put together such a thoughtful study that made for such compelling results. I wanted to thank the study nurses. This is really a ton of high quality data, and it has been a privilege to work with them. I also want to thank Damaris Kamonge for helping support with the data processing, and Rita for keeping for the administrative support and keeping everything together. So thank you to everyone.
Thank you very much, Molly. Yes, Molly. We'll now have Dr. Shavin Jacob. You can make your comments.
Yeah, thanks. I don't have much more to add. Just to sort of echo the sentiments of Beth and Molly in terms of just the admiration for the team for pushing through and getting this done at KNH, and you know, sort of just also to kind of urge fellow clinicians to kind of think about you know, what is it that we can do to sort of optimize safety and quality of care for our patients. And you know, as you sort of reflect more on the DripAssist and what its use case has been in terms of this study, sort of thinking about, you know, other use cases that you may require for the setting in which you work.
Thank you. Thank you, Dr. Jacob. Dr. Irungu Kamau, please give us a closing comment. Sorry, Dr. Joyce Swamisha.
I don't think I have anything more to add. I just would wish to encourage the team to consider scalability, but before that, even for purposes of awareness, let's do an evidence brief as fast as possible and get it out there through the various portals that are there. Thank you.
Thank you, very important. Dr. Kutoi, welcome.
I think a big thanks to the participants who joined in today, and also for the team that I was glad to be part of, and Shift Lab for giving us the opportunity to prove the concept within our Kenyan labor ward. And as from the question, realize this is about infusion, so it can be used in any other infusion. So we hope that at the level of scalability, even pediatrics and the other commoner places where infusions are made could be included. It was a pleasure. Thank you.
Thank you very much. Mokwana, welcome. You can also give us your closing comments.
Thank you very much for everyone who participated in this webinar. And also thank you for the team that I worked with, especially the study nurses who worked tirelessly and very hard in the labor ward. And as a clinical nurse, I would want to say that this is a device that every midwife would wish to have in their facility. It is easy to use. You know, it's something that will give you a very easy time, as Beth had told us. It's something that will alarm you when something has gone wrong. And as I said earlier, our workload is not small. We have very many patients, and you can imagine if you have half of the labor ward is on drips, you need something that you're comfortable, that the drips are running well at the right rate. So I just wish this is something people would buy in and make it very easy for nursing care of our patients. Thank you.
Thank you very much for that. Thank you, our participants. We've also had questions about PPD, and I'll invite Dr. Ewak to give a comment, especially to the pharmacists who are really asking about their CPD points and how to access that. Dr. Ewak, welcome.
Okay, thank you very much, Dr. Karume. Now, with regards to the queries regarding CPD, on Thursday, just 30 minutes before the session, I believe the session will be at 7 p.m., so around 6:30, we'll be able to have a team, or at least one representative from the Pharmacy and Poisons Board, Dr. Lily Bett, to be able to address some of the CPD concerns we have. And for those who have challenges navigating the PPB portal, she'll be able to take us through that. But in terms of those who are not able to see the points, we send it as a mass mail. So you may need to check your promotions or spam folder, or if you have whitelisting software on your email, you may have to add cpdknhpharmacy@gmail as one of your contacts. That could be a reason why you're not seeing the emails that we sent to you. But otherwise, the points are sent like about 2 weeks after the event. And for the events, because our events are numerous and we are not able to upload them on the portal before they happen. So keep looking for the events even 2-3 days after it has happened. By then it should have already gone through the 2-step approval process from the board for you to be visible, for you to be able to subscribe. Thank you.
Thank you very much. For the others, if you have any question, you can also email the knhcpd@gmail.com. And we really want to thank our presenters for the person supporting the study, and just for joining us for this session. And even as we come to towards an end, Dr. Kinuthia, you can wrap it up for us.
Yeah, thank you, Agnes. I don't really have much to add. Is to thank everyone who has joined and to invite you for the coming webinars. Thank you.
Thank you very much. Indeed, you're welcome to join us for our coming webinars. We'll have one at the same time tomorrow from 2 to 3:30 on neonatal sepsis. A very interesting one with regards to vaccines on Thursday from 7 p.m. to 8 p.m. And as you heard, you can join in earlier 6:30 p.m. to hear from the Poisons Board. Thank you very much, everyone. Do have a lovely afternoon.